start with a sound check. Can you hear me in the back row there? Can you hear me in the back row there? Okay. You may have already looked at the ICON website to notice that we have got back the statistics on the test. And the questions, as a rule, were all sound questions. That is, students who knew other things on the test could answer those questions. But some of them were harder than we intended to make them, as can happen. And so the thresholds for this exam are going to be set down 10 points. So someone who got 80 points on the test or above would have an A for that test. 70 to 80 would be a B. 60 to 70 would be a C. And at the end of the course, the final thresholds for the different grade points will be settled. Is there a comment you'd like to make? Is there a comment you'd like to share with us? Ah, okay. Well, we all have those, you know. So, let's see if we can get that to open. Open, there we go. There we go. So I know we finished citric acid and that we've had the exam on it, but having had the exam, do you still have any questions on the citric acid cycle that you'd like to ask? Okay. So in thinking through carbohydrate metabolism, glycolysis, gluconeogenesis, the citric acid cycle, we've started with kind of the core of metabolism. But so far, everything that we've done has been kind of the baseline behavior. It doesn't take advantage of this incredibly powerful molecule out there, oxygen. And what we're going to be talking about today is the set of reactions and structures that make possible a process called oxidative phosphorylation. And it's oxidative phosphorylation that makes interesting life possible, as opposed to just life. Because until you get to this level of energetics, you don't have enough energy to do very much. So what does oxidative phosphorylation actually do? Let's take a look at that. And here we have our high-view roadmap, you know, the MapQuest version of metabolism. And we've done citric acid cycle. We've seen how feeding carbon intermediates into that can produce NADH, you remember. And so we've been accumulating NADH left, right, and center, as if there was something nice and useful about NADH or QH2. And oxidative phosphorylation is going to be the process that's going to use the electrons that have come from a variety of fuels, all kinds of fuels. It's going to use those electrons to create a proton gradient, to create a concentration of protons on one side of a membrane that's greater than the other. And that proton gradient is going to be the thing that lets the cell make ATP. This is not at all an intuitive concept. Substrate level phosphorylation, when you make an ATP from phosphoenolpyruvate, makes sense. Making it off of a proton gradient, not as obvious, but incredibly cool. And what this allows us to do is to have a common pool of energy that serves all the different needs of the cell, because all the different fuels can be fed into oxidative phosphorylation and generate this common pool. So we're going to have some reduced compounds. NADH, FADH2 are both reduced compounds. Those reduced compounds come to us from, for instance, glycolysis. You remember glyceraldehyde phosphate dehydrogenase. We're going to see that we can get them out of fatty acid oxidation in a little bit. We've seen us getting NADH from the citric acid cycle. And these are fairly rich, energy-rich compounds in their own right. They also have electrons that have a high transfer potential, a high potential to be transferred to other molecules. And we're going to see why that's a useful thing. It's specifically oxidative phosphorylation as contrast to substrate level phosphorylation that we've already seen, or photophosphorylation that we'll see in photosynthesis, because we're making ATP, that's the phosphorylation part, by transferring the electrons from these energy-rich compounds to oxygen. So we're going to use oxygen gas as the final electron acceptor. And Isaac Asimov once described the capacity to use oxygen this way, that if you're going to make garbage, you might as well burn the garbage and stay warm by the fire. And that's sort of the underlying principle of oxidative phosphorylation. This is going to happen because of electron transport in the inner mitochondrial membrane. So we're going to see that this inner membrane is very well organized. By generating a proton gradient, that stores chemical potential, and then the ATP synthesis occurs when the protons flow down the gradient back to where they came from. And for all of this to make more sense, you need to start to get to know our friend the mitochondrion. Mitochondria are small double membrane bound organelles present in almost all eukaryotic cells. And they're often depicted as being ovals. They do come in other shapes as well. And they have a lot of architecture, and that architecture is associated with specific kinds of metabolic specialization. So there's an outer membrane, which is yellow here. There's an inner mitochondrial membrane, and you can see in this drawing of it how that inner mitochondrial membrane is folded into lots of little folds, sort of like you take a balloon and then squish it together. Or maybe if you've done that trick with drinking straw wrappers and you squish it together. That's the kind of image you should have. And those squished in parts, the little invaginations, are called cristae. Singular is crista. Between the two membranes, you have an intermembrane space. So there's an actual space between the two membranes, and that's going to be a place where important things happen. And then inside of the matrix, inside of the mitochondrion, filling the rest of the interior is stuff called matrix. Matrix is from the Latin for mother stuff. This is sort of the mother stuff of the mitochondrion. These different regions are all associated with different metabolic activities. So for instance, the citric acid cycle happens here in the interior in the matrix. We're going to see fatty acid oxidation happens there also. In the inner mitochondrial membrane, there's the respiratory chain, the integral membrane proteins that handle the electron transfers for oxidative phosphorylation. And it's as those electrons are transferred that we generate the proton gradient. And this is an extremely schematic one. This is the inner membrane here. Okay, this is the inner mitochondrial membrane. So what the electron transport chain's job is going to be is to move electrons, and by transferring electrons, move protons out, generating this gradient of positive relative to the inside. And then here, in a separate step, we're going to have protons flowing back in to generate ATP. This is an ATP synthetase, so it makes ATP. And we can use oxygen as the electron acceptor. So you can see here's an oxygen molecule with electrons added to it, and picking up the protons, you're making water molecules. Very dangerous business working with oxygen. We're going to see how the extreme precautions that the cell takes to handle those molecules of oxygen. So to think about the electron transport chain within oxidative phosphorylation, we need to remember or learn some things about redox reactions in general. We've referred back to some of this, but this is a good time to refresh all of that. So there's a whole category of reactions that we call oxidation reduction reactions, or for shorthand, redox reactions. And they are characterized by the fact that electrons are transferred between molecules. Reduction is gaining electrons. This is a little counterintuitive. If you add an electron to something, that reduces the molecule or the atom. And that's because electrons are negatively charged, and it reduces the valence state of that atom. So if you start with an atom of iron 3+, and you reduce it with an electron, you now have Fe2+. So this iron atom has been reduced. Its valence state is lower because it has more electrons, which have negative numbers. When we are speaking about the things coming up, we're going to be talking about reducing agents and reductants. So a reducing agent is a molecule that can add electrons to another molecule. And when the reducing agent or reductant does that to another molecule, the reducing agent becomes oxidized. If something else is getting reduced, this has to get oxidized. And then the opposite side of that is the oxidizing agent or oxidant can pull electrons out of a molecule, and when it does that, the oxidizing agent gets reduced through the reaction. So this is a reaction that we've seen before, pyruvate plus NADH going back and forth to lactate and NAD+. I think the next one repeats that here. So here is this familiar reaction, and I'd like you to chat with each other for a moment and see if you can agree on which things are the reducing agent, which ones are the oxidizing agent, which molecules get oxidized, which molecules get reduced. Okay? And you can take a look at the picture if that'll help you think about it. Anyone want to venture anything on who's doing what to whom, with what, where, and how often? As the old question goes. Well, let's take a look at this. This particular redox reaction has a kind of a handy clue in it because NAD and NADH are so commonly used in reactions like this that we can get used to seeing them. So is this the oxidized or reduced form of NAD? Reduced form. This is the reduced form. This is the oxidized form. Okay. So if this is the reduced form, it can act as a reducing agent and reduce pyruvate. So if it reduces pyruvate, then that molecule becomes lactate, and the NAD is now oxidized to NAD+. So this is the oxidized form. This is presumably the reduced form compared to pyruvate. And let's take a look at the molecule and see if that makes sense, okay? So we said based on what we were seeing with the NAD, where this is the reduced form, this is the oxidized form, if this is going to act like a reducing agent, this should be a reduced molecule. We look at this carbon here, and we see that this carbon has one bond to an oxygen and one to a hydrogen. This one has two bonds to an oxygen. This is a relatively oxidized form and a relatively reduced form of the molecule. So NADH is going to reduce that carbon down to that, and now we have lactate. Okay? And the contrast here between the Fe3+ and Fe2+ is that in covalent molecules where there's a big difference in electronegativity between the two atoms, if one atom is more electronegative, it's considered to have the electron. So with a carbon-hydrogen bond, carbon wins the electron. In carbon-oxygen bond, oxygen wins the electron. Okay? Any questions about that before we move on? Okay. All right. These are just a couple more examples to help you get a little bit of a feeling for what's going on here. So in hydrogen and fluoride, you can start with gaseous hydrogen and gaseous fluorine. In the molecule hydrogen fluoride, fluorine tends to win that electron, so the fluoride is relatively, the fluorine atom is relatively reduced, and the hydrogen atom is relatively oxidized. Here's a molecule that we all need. Some of us don't get enough of it. It's vitamin C. And so here you can see in the reduced form of vitamin C, it's got hydrogens there, bonds to hydrogen, a bond to a carbon. Here's the oxidized form, and you can see that carbon has now been oxidized relative to that one. So vitamin C can serve as an antioxidant. It can go in and reduce other compounds as it becomes oxidized. Saves your gums. Now, this notion of giving up electrons and so forth, you might think intuitively that not all molecules are equally good at doing this. And you'd be right, that whenever you have a property like this, there's going to be some that do it more and some that do it less. And you might wonder, how would you measure something like the tendency to reduce other molecules? We have a property we call reduction potential. It's called E prime sub 0. The reduction potential, the redox potential, the oxidation reduction potential, these are all synonyms. And in order to find a number for a redox potential, we talk about couples of molecules at a time. So there's a redox potential associated with a redox couple, in which X is the oxidized form of it and X minus is the reduced form of it. So you're always talking about a redox potential of a couple of molecules. And we do this by having a reference cell. So you have one side of a cell that has 1 molar solutions of X and of X minus, the reduced form. And we are going to compare electron movement to that of a standard cell that we've chosen to be, we just decided to do this, hydrogen as in the form of H plus and H2. So this would be the oxidized form, and then this would be the reduced form. And then by having an agar bridge between them, we keep the molecules from moving in bulk, but we can look at electron movement, and we can pick up a voltage difference between these two cells. If the solution we're testing is a better reducing pair, electrons can flow over. If it's better at oxidizing things, it can grab them away. So for a setup like this, the overall reaction would be X minus, the reduced form, plus protons, go to X, the oxidized form, plus a half a molecule of hydrogen gas, which we can break out to these half reactions, X minus, X electron, H proton plus electron to a half H2, but the electrons are not free in this process. And the way of thinking about this is that the larger the redox potential, the more likely it is that the oxidant will tend to accept electrons at pH 7. So a large E0 is going to make it likely that the oxidant will accept electrons, will oxidize other things. And we can get a little bit of a feeling for that by looking at some standard reduction potentials. I do not expect you to memorize these reduction potentials. Okay? These are examples to give you a sense for the fact that the variety of biological molecules differ in their ability to steal electrons or to give electrons to other molecules. And for redox pairs that have a highly negative electron potential, the reductant of the pair, in this case alpha-ketoglutarate, is going to tend to give up electrons. It's going to tend to reduce other things. Whereas the further you go down to the positive redox potentials, the more likely it is that the oxidant is to pick up electrons, is better at oxidizing other things. Sorry. And we're going to see that oxygen, while it's an absolutely wonderful oxidant, is actually not the very best oxidant in the biological world, but it's a very, very good one. And it's because it's such a strong oxidant that oxidative phosphorylation is such a powerful way to get energy out of biological molecules. Okay. All right. So we've talked about how redox reactions are actually the sum of two half reactions, so that if we wanted to talk about pyruvate and NADH going to lactate and NAD+, you can break that into two pieces of pyruvate plus 2 protons and 2 electrons go to lactate, because that's what it takes to make pyruvate into lactate, and that NAD+ plus protons, electrons go to NADH. Some of the interesting properties of electron movement and transfer is that it is easier to move electrons through proteins to a point that we can pull a field across that, and more electrons will be moving there until we start to get such a field that they're kind of crammed up at the door, like the people trying to get out of a burning theater. So it's not a simple linear relationship. It's one with an optimum. Well, I think we have enough of the reference terms now to start thinking about oxidative phosphorylation, and specifically about the part of oxidative phosphorylation that we call the respiratory chain. And the respiratory chain is going to be an electron transfer chain that's going to have the ability to pump protons. So the respiratory chain has 3 proton pumps, and it has one complex, complex 2, that is coupled to the citric acid cycle. All of these complexes are involved with moving electrons between different molecules. The proton pumps, complex 1, complex 3, because complex 2 is succinate Q reductase, and complex 4, to connect the different proton pumps, we need electron carriers of some kind or another. And we have Q that carries electrons between complex 1 and complex 3. Q also carries electrons from complex 2 to complex 3. And we have cytochrome C that can carry electrons from complex 3 to complex 4. And at complex 4, those electrons are going to have to finally be dumped onto something, and they're going to finally be dumped onto oxygen and make water. So oxygen is the terminal electron acceptor in oxidative phosphorylation. Any questions? Okay. Eli, I think we should hand out some file cards. So if you'd like to let me know that you're here today, you're invited to do that. So if you have comments you want to let me know, you're welcome to use a card for that purpose, but otherwise just let me know. If you need more cards, I have them here, Eli, okay? So I don't think we need to worry. You can do that and pay attention to this at the same time. This is to help make sure that you're oriented as to where all this is going on. This is another representation of a mitochondrion. And you notice here the space between the outer membrane and the inner mitochondrial membrane. The matrix fills the deep interior, and that intermembrane space is going to be a place where exciting things happen. This kind of sausage-shaped mitochondria is a very commonly represented one. But in fact, mitochondria take on lots of different shapes, and this is a couple of different examples of it. Here's a mitochondrion that has been reconstructed by serial electron microscopy, and this is the mitochondrial network in a fibroblast growing in tissue culture. Cells can actually move mitochondria to the place in the cell where there's the most demand for energy, so the location can reflect energy needs. But however they look on their outer shape like this, whether they're nice little sausages or peculiar networks, they all have these invaginations of the inner membrane that we call cristae, or that one would be a crista. We saw that we can make some NADH inside the mitochondrion in the matrix through citric acid cycle. Some of the NADH is made out in the cytoplasm through glycolysis. How did that NADH get into the inside? And it turns out, like so many things, there are transporters that can accomplish that. But we're not just going to let it flow in, or the NADH from the inside would flow back out when we make it in the citric acid cycle. So we have to get it inside some other way. And this is the malate aspartate shuttle, which is one of the ways in which mitochondria can move reducing equivalents without moving the molecule. So out here in the cytoplasm, I don't like calling it cytosol. It's cytoplasm. There's nothing sol about the cytoplasm. Good, long history. If people want to know, I'll tell you. So out here in the cytoplasm, we have NADH that we've made, say, example, through glycolysis. We want to get it into the mitochondrion. Don't want it to leak in. That would let others leak out. So a molecule of oxaloacetate in the cytoplasm can be reduced to malate. Here's the carbonyl oxygen. Here's an alcohol. So this carbon was reduced. So the electrons on the NADH are now on the malate. The malate can go in through the malate shuttle. Malate in the mitochondrion, of course. No problem there. We've got malate dehydrogenase. We make an oxaloacetate recovering our NADH. And then the oxaloacetate, through the transamination reactions we mentioned earlier, can be made into aspartate, an amino acid. So here the amino acid is acting as a carbon backbone to carry electrons. So here we have aspartate. It's going to go out into the cytoplasm. Aspartate can be used to regenerate the oxaloacetate in the cytoplasm. So these two enzymes, although they're both called malate dehydrogenase, are in different locations. They're different proteins. They're what we call isozymes. That in the body, one of the ways that accomplishes the job of metabolism is by having enzymes that have certain specific properties on different parameters. And this is one of the ways that the cell can get the electrons from NADH into the matrix without leaking stuff. Another example of something that's got to be moved around has to do with ATP. Most of the ATP that's going to be needed is going to be needed in the cytoplasm. Most of the ATP that's made is going to be made in the mitochondrion. And even to make ATP, you need ADP, and ADP is generated in the cytoplasm. So we're going to have to get stuff to move between these two compartments. And we see transport molecules in which the ATP can enter from the matrix side and be released on the cytoplasmic side. And ADP can bind and enter the mitochondrion. And we don't need to expend any energy to make this specific. If it binds both ATP and ADP equally well, since there's more ATP here and more ADP here, they're going to go in the right direction because they're flowing downhill. However, if you think about it, there's to make an ATP into an ADP, you're going to need some inorganic phosphate. And so in addition to the ATP or ADP shuttle, you have a phosphate transport system that lets phosphate flow into the mitochondrial matrix as well. Okay. So let's start talking about the components, the different substructures within the electron transport chain. We mentioned complex 1 was the first proton pump in the electron transport chain. And it looks kind of like this. It's kind of strange to me that we can actually talk about the shapes of these complexes, but there you are. This is an example of complex 1 from one particular organism. And within this complex, we're going to have a number of different redox partners taking a role for all this to happen. One of the common things in respiratory chains is iron-sulfur centers. So you notice here you've got iron-sulfur to iron to sulfur. So this iron-sulfur center is alternating iron-sulfur atoms that are capable of having multiple redox states. Here's a simpler iron-sulfur complex with just 2 of each. And an important carrier is a flavin mononucleotide. So this is a flavin ring. Again, we saw the flavin ring back in pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. So we know that it's a redox compound. But it's mononucleotide because it has only a single ribose ring. So this is does not have any adenine on it anywhere. And so these conjugated double bonds can receive electrons and be in the reduced form over here. And they've received the electrons from the NADH. If we want to look at this a little more schematically, here is complex 1. It's an integral membrane complex. It's multiple proteins. It's not just one. The NADH is being generated or is approaching the complex from the matrix side. And there are protons out there as well. And in reacting with complex 1, the electrons from the NADH are transferred through these iron-sulfur centers and flavin and so forth, ultimately onto a molecule of ubiquinone. So complex 1 accomplishes the transfer of electrons from NADH to Q to make the reduced ubiquinone. Associated with these electron transfers, sort of through the machinery of complex 1, protons are moving from the matrix side out into the intermembrane space. So we're starting to build up a proton gradient. There's some evidence that suggests that to get those protons from here to there are something called a proton wire, where the structure is just lined up so perfectly that the protons just jump right on down it. But you can imagine that that's not a trivial thing to move protons around. The protons that are moved are not necessarily the protons that were up here. They're just taken from the solvent phase of the matrix. So now we've got a molecule of QH2. What should we do with that molecule of QH2? Okay. Well, actually, we can get more molecules of QH2. This is just a reminder that succinate dehydrogenase is an integral membrane protein. It's part of the citric acid cycle, but it's also an integral membrane protein. It's a place where the citric acid cycle is anchored into the inner mitochondrial membrane. It was taking electrons from succinate and is going to transfer them to ubiquinone. But it does not pump protons. It can transfer electrons, but it can't pump protons. So this is just an electron transfer machine. Some other places where we can get electrons into the electron transport chain is through what's called the glycerol phosphate shuttle. If we don't have very much aspartate, we can still get electrons from NADH from the cytoplasm into the mitochondrion. The electrons from the NADH can be used to generate glycerol 3-phosphate. That can move through the membrane in the mitochondrial matrix. Mitochondrial dehydrogenase takes those electrons to make QH2. And then the dihydroxyacetone phosphate, so it's the more oxidized, this is oxidized here, can go back out into the cytosol, pick up another pair of electrons. So a couple of, probably 3 different ways at least, in which we can feed electrons into Q to make more substrate for the chain. But neither of these guys can pump protons. They're just in the electron transfer business. Now we're down to complex 3. So the main chain goes complex 1, complex 3, and then complex 4. And the job of complex 3 is to take the electrons that are on ubiquinol, or QH2, and get those electrons onto cytochrome B and C. The cytochrome B iron is going to be reduced, and there are also other things in the complex like cytochrome C1, iron sulfur centers, lots of good redoxy juiciness. Here's heme B. You notice there's a heme ring here. That heme ring looks a lot like the heme ring of hemoglobin, but in this case that iron does go through redox cycles. One of the jobs in hemoglobin was to protect the iron from oxidation. This one likes to switch back and forth, Fe3, Fe2. It has a little bit of spinach hanging off its sides. So there's the heme B from cytochrome B. The cytochromes have their names because having a heme ring in them with the iron, they all are colored. So they're cytochromes. They're colored molecules from the cytoplasm. Early biochemists were so creative. So here's a picture of complex 3. This is, again, an integral membrane complex. It's got multiple polypeptides in it. There are prosthetic groups on that complex that are beautifully set up for picking up the electrons and transferring them to the next recipient in the chain. But there's a little bit of trickiness because ubiquinol, the reduced form of ubiquinone, has 2 electrons on it, and cytochrome B can carry 1 electron at a time. So we've got to work out a way to shovel or to funnel 2 electrons down to 1 molecule some way. And that's accomplished through something called the Q cycle. And this is what the Q cycle looks like. So here we have complex 3. And we're going to start here with a molecule of QH2. And it's going to donate its electrons. Remember, it has 2 electrons. It's going to donate its 2 electrons. One of those 2 electrons will go to cytochrome B, and another one of those electrons will go to an iron sulfur center, or an iron sulfur protein. That gave us a molecule of Q again. Let's follow those for a little bit. So the electron on the iron sulfur protein can be transferred to cytochrome C1, and then that can be transferred from plain old cytochrome C3+ to make it into cytochrome C2+, which is the reduced form. But here we have cytochrome B that's still integral inside complex 3, and we're not quite done with the dancing around. Cytochrome B is able to transfer its single electron to the plain old Q to generate a half-reduced semiquinone. So that's why it has a dot on that. That's telling you this is semiquinone. It's only halfway there. In the second round, a different molecule of QH2 coming from somewhere, again gives up an electron to cytochrome B, again gives up an electron to the iron sulfur center. The iron sulfur protein is going to transfer it through C1 and make a second molecule of cytochrome C2+. Cytochrome B now has that electron on it, and it can finish reducing Q back to QH2. And in doing that, we're picking up protons out of the matrix. And so that helps to generate that proton gradient. So it's a little bit indirect. Because ubiquinone can carry 2 electrons, and all the other carriers inside of the complex 3 carry 1 at a time, it has to do 2 rounds of reduction to get the transfer completed. So the overall effect of the Q cycle is we get 2 molecules of cytochrome C reduced. We get the 1 molecule of ubiquinone oxidized, and we move 4 protons altogether into the intermembrane space. So that's cytochrome 3. That's the way it accomplishes the proton pumping. So we've got electrons moving. We've got protons being pumped. So we're still building up a proton gradient in this intermembrane space. Okay. So we had ubiquinone, which was lipid soluble, and it just floated and floated from complex 1 to complex 3, or from any of the other things that make ubiquinone. Cytochrome C is a protein. It's not a lipid, and it is a peripheral membrane protein. If you look here at this molecule, these are all negatively charged groups, and it rolls along the surface of the membrane. So it's a very important peripheral membrane protein. It's on the surface of the intermitochondrial membrane, carrying electrons from complex 3 to complex 4. We can tell that it's an extremely important protein and that it's really good at what it does because it's very highly conserved. Cytochrome C is one of these very highly conserved proteins that there just aren't a lot of other ways to do what it does. So complex 4 has maybe the hairiest job of all. Complex 4 is going to regenerate cytochrome C by oxidizing it. So it's going to steal the electrons from cytochrome C. And it's going to transfer electrons to oxygen to make water. But it takes 4 electrons to completely reduce an oxygen molecule. So somehow or other we've got to take 4 cytochrome Cs and reduce 1 molecule of O2. So it's going to be a challenge. This is a representation of complex 4. Again, it's an integral membrane complex with actually quite a number of different proteins in it. We call it cytochrome C oxidase because it's going to oxidize the cytochrome C that was reduced before. The electrons from cytochrome C are going to be moved through various locations within cytochrome C oxidase. Copper is going to be important. Heme A. There's a special funky thing called a binuclear center that is going to be able to handle things that are very reactive and potentially damaging. This is a proposal for how on earth you can handle something as explosive as oxygen and not wipe out the proteins in the process. Oxygen, we know, is great at burning things. How do you handle it in a controlled way so that you don't have other things being damaged at the same time? And one way that it works is that the oxygen is always attached to something, and the electrons are always under control. It's going to use both iron and copper to do this, and it has to take multiple steps to do that because you remember you get 1 electron at a time from cytochrome C, and you need 4. So let's start with 1 molecule of O2, and it's going to bind at the iron-copper center, interacting with the Fe2+. So we have 1 molecule of O2 bound here. We start feeding in electrons that are coming from cytochrome C, protons from wherever. And you notice that this molecule of O2 has been split into 2 oxygen atoms, right? And this one has been slightly is starting to pick up electrons. Another proton can come in. This OH can come off as H2O. We still have the other oxygen back there. We need, I should maybe not duplicated that there. So we now have the O2 minus sitting on the iron 4. We pick up more electrons. And if you notice here, iron 4 is going to iron 3. And the oxygen is going from 2 minus to plain old OH, picking off that proton. And we repeat the trick of pulling in more electrons, and we come out as water. So it's the oxygen atoms are never free to go around and do things, and they're always under control. And in the process, we're moving a fair number of protons from them out of the matrix into water atoms, using them up to make water molecules. Sorry, using protons up to make water molecules is also creating a proton gradient if there are fewer protons left in the matrix. So one molecule of oxygen here has made 2 molecules of H2O, so that's moved 4 protons effectively out of the matrix. So this is sort of the grand overview. I'm sorry, it's a little not too bad up there. So here we have complex 4. Cytochrome Cs come up to it in their reduced state. They transfer their electrons to complex 4 and go back to as cytochrome C3+, so they can go back and get more electrons. And it can catalyze this transfer of electrons to attack the oxygen atom and release as H2O. So we have reduced water, or oxygen has been the oxidizing agent. And in the same time, protons are accumulating on the intermembrane side. This one may also have some proton wires. And so in addition to the protons that are used up to make water, there is some proton movement through the effect of the electron transfers in the interior of the complex. Any questions so far? Any questions about the electron transport chain? This intermitochondrial membrane thing, you've got big complexes that can do this kind of scary work of moving electrons around, moving protons in combination. Any other questions? Well, how much energy can we get out of this? And one way to think about that is looking at the delta G associated with these reactions. And so the net reactions at complex 1 have a delta G of minus 70 kilojoules. Complex 3 has minus 37, and complex 4 has minus 112. Oxygen's powerful. But each of these complexes is enough to account energetically for making an ATP. They don't make ATP themselves, but they have enough energy happening to accumulate the energy to be used to make ATP. So each complex can account for an ATP molecule energetically. A lot of time was spent trying to do it at the molecular level, and that turned out to be a goose chase, a wild goose chase. I've talked about these complexes as if they're all free floating in the membrane, but in fact, in many mitochondria, the different complexes are associated with each other in order to make it a more efficient process. So for instance, here's a complex 1 that's interacting with a complex 3, and that complex 3 has a couple of complex 4s attached to it, so that you can end up transferring those electrons just right down, flowing on down. So the thought is that these complexes of these assemblies of complexes allow for a more efficient transfer of the electrons. It's called the respirosome. So all we've done so far is move protons around. You know, we're starting to pickle the intermembrane space. But that by itself is not ATP. So somehow or other, we're going to have to turn a bunch of protons into some ATP. And this is accomplished by something called chemiosmosis. All of this moving of protons from the matrix, the most interior part of the mitochondrion, into the intermembrane space has created an electrochemical gradient. So it's an electrical gradient because there is actually an electrical charge difference across it. And it's a chemical gradient because there's a concentration difference across it. And so for 2 electrons that are fed into the electron transport chain, roughly 10 protons are moved. And this insight was made by a British scientist called Peter Mitchell. That up until the work of Peter Mitchell, people tried really, really hard to find the high energy intermediate that was transferred between complex 1 and complex 2. And they looked and they looked and looked. They never found a high energy intermediate that was transferred between these complexes. And Peter finally said, you're thinking the wrong way. It's not a little molecule to look for. It's a property of the whole mitochondrion. It's not any one thing you can pull out. You have to use the whole mitochondrion, and what's happening is the mitochondrion as a whole is sustaining this electrochemical gradient, and it's going to use that to generate ATP. It's a very important concept to get a hold of, not just because it keeps you alive, but because we're going to see this again in photosynthesis. So this concept of chemiosmosis and electrochemical gradient is going to turn up again in photosynthesis. So maybe we should call that quits for now. Have a great spring break. Good to see you.